Hi there, this topic is topic 17, it's inheritance. And for this part we're going to look at inheritance 17.1 and 17.2, which is talking about chromosomes, genes and proteins. The first thing we've got to look at is defining inheritance. So inheritance is simply the transmission of genetic information from generation to generation. So taking those hereditary traits like eye colour or hair colour and passing them from parents to offspring. Okay, so the next subtopic is quite a lot more we need to talk about, but it's all very simple. First thing we look at is a chromosome. Now, a chromosome is here. This is a chromosome. It's made of two chromatids. Chromatids are joined together with a centromere. Now, a chromosome is just a strand of DNA. And all it's done is it's coiled up. So the DNA will be, if it was to unwind, it would be all filled in here, and you could pull it out and it'd stretch on for miles and miles. But a chromosome is a coiled, tightly wound and packed part of DNA, and it'll have lots of different genes in the chromosome. There you can see a gene for hair colour, but there'll be a gene here, 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 and all the way through the chromosome. You can only really see DNA or chromosomes when they are coiled up as chromosomes just before the cell would divide. So this happens in a process called mitosis, which we'll talk about later, and meiosis. But yeah, you can't really see chromosomes normally in a cell. You can only see them when they divide. Somatic cells here, it says, have 46 chromosomes. These are your normal cells. It's 23 pairs of chromosomes. So if this is one chromosome, there'll be another X here, another chromosome, and that would be a pair of chromosomes. So you have one chromosome, two chromosomes, that's a pair. So you will have 23 pairs, but you will have 46 of this image here. Now, this is called a human karyotype. Now, a karyotype shows all the chromosomes from an organism's DNA all next to each other lined up. So as you can see, chromosome, chromosome, pair of chromosomes. The chromatids here make up each chromosome. There's two chromatids. Now, all chromosomes have a matching pair. So for example, this chromosome here will have a matching pair right next to it. One will be from mother and one will be from the father. And the same here. This one will be from mother, this one will be from father. They will contain the same genes and they'll all match up. So if this is the chromosome that has the gene for hair colour and it's right here where the arrow is, then on the mother one it will be right here as well. And yeah, so that's a karyotype, and they all have a matching pair apart from this one. This chromosome pair here determines sex, so like male or female. These don't match. These are not matching pairs, which is why it determines the sex of the offspring that's made. The next two things are fairly easy. We've got to define a gene and define an allele. Now, a gene is just a length of DNA that codes for making a protein. So here you can see a gene, and this is just a section of the DNA that's all coiled up here as the chromosome, and it will code for the making of a protein. Now, we've looked at DNA structure before. It's made of nucleotides and bases. Those bases code for making amino acids. Those amino acids would join together to make proteins, so that's what the genes are there for, and that's what they do. Now, alleles are basically, it's a version of two options of a gene. So if you have a gene here for hair colour, it might be brown. It might make a brown hair colour protein. On the other one, there'll be a matching pair, a matching version allele, and this will be your other option, and this might be blonde hair. And whichever one is more dominant will be the one that's expressed in the gene and make that certain type of protein. So alleles are just the versions of the gene that are available. You get one from mom and one from dad. Okay, objective D is to describe the inheritance of sex in humans. So are you going to be male? Are you going to be male? Or are you going to be female? Now, it's pair 23, so 21, 22, 23. This is the pair that determines sex, whether you're male or female. Now, XY would indicate male, and X, so this is a male karyotype, and XX would be female. If I use this diagram to explain it, a male is XY and a female is XX. In her cells, she will have all these chromosomes, or he will have all these because this is a guy, and then the female will have all these as well, but XX at the end here. When they undergo meiosis, meiosis is about making gametes. It's about making sperm cells and egg cells. So when they do that, one of those goes in, who knows which one, one of those goes in, one of those goes in, one of every pair goes in, and you end up with a cell, a sperm cell or an egg cell, with 23 individual chromosomes. Now, that cell could go in, that chromosome could go in, the X chromosome, but also the Y one could go in. We don't know which one's going to go in there. So males will have sperm cells, gametes, with either an X in or a Y in. And females, all her egg cells will have X's in. Now you might look at that and go, well, doesn't that mean there'll be more females in the world rather than males? Because there's only one Y chromosome there in the male sperm. If you do a monohybrid cross, which is just showing a diagram of how gametes could fuse to create different variations of offspring, you can see that the ratio is 50. The ratio is 1 to 1. The percentage chance of getting a male to female is 50%. So what you would do here is, this is not if a male and a female have copulation and reproduce to make offspring, they're going to have 4 children. This is just showing the chances of those offsprings being male or female. So this gamete here from the male, because a male will only have X and Y. This gamete here, if it's an X that comes into contact with this X, this will make an XX diploid zygote. Now obviously we're not looking at every single chromosome here, we're just focusing on this one. If we were to draw every single chromosome out on this diagram, it would be chaotic, so we're just looking at pair 23, because this is what determines sex. So X joins up with X, and we've made a female. And you just follow that through. This X joins with that X, you've got a female. But whenever there's a Y, there will be a male. So as this sperm gamete joins with these two X's, it's going to make XY, XY. And as you can see, it's a 1 to 1 ratio, 50% chance of getting a male or female. This is a little picture to show you how gametes are made. So as you can see, here's the chromosomes here. This is an ovum producing cell. It's diploid. It's got all 23 pairs of chromosomes, 46 in total, and as you can see, it's a female. It's got the X and X chromosome, both the same size. And when the ovum producing cells make the ovum, or the ova, plural, they will take 1 and make haploid cells, which are the ova, and they will have an X chromosome and an X chromosome in there. With the sperm producing cells, the cells in the testes that produce sperm, they will have a diploid chromosome amount, so they'll have X and Y. Y is shorter, it's not as long as the X, which is very important, and we'll come on to that later in the topic. But yeah, when they create haploid cells, the sperm cells here, an X will go into one and a Y will go into the other. And yeah, that's making haploid cells by the process of meiosis. Meiosis only occurs in the body in the gonads, which is your testes if you're a male and your ovaries if you're female, and it's all about making gametes. Meiosis, gametes. So objective E is to explain how base orders in DNA code for amino acid production. Now, if you remember before, this is the nucleotides we were looking at. It's the sugar, phosphate, backbone, and it's got sugar, phosphate, and a base in it. This is one nucleotide. This is several nucleotides, and this would form one strand of DNA, and then you'd have like a mirror here effect, and you'd have the bases here with the sugar, phosphate, backbone there, and that would make a double helix of DNA. So each nucleotide has one of the 4 bases, as we remember, A, T, C, or G, and a string of nucleotides would have lots of different bases. As you can see here, here's the sugar phosphate backbone of the DNA, and here's the bases it has, C, A, T. Every three bases will create one amino acids. That will code for an amino acid production. CCA will make glycine. CGA will make alanine. When you join these amino acids that are made, you will make a peptide. Now, a gene is when a full protein is made, not just like a small peptide, but a full protein. So when you join hundreds of these together, of these amino acids, with hundreds more of these bases, you will make eventually a protein. And once you've got a code for a protein, like this would be the code for the protein that would be made here, that would be called a gene. So the gene we looked at over here, this gene here, if we zoom in, that would be hundreds and hundreds of these, coding for hundreds of amino acids, which join together to make one protein. That's what a gene is. So if these amino acids join together to make insulin, which is a protein, that gene would be the gene that codes for making protein. And that's how it all works. Like we said earlier, insulin is a small protein. It's made of 153 bases here. So 153 of these will code for 51 amino acids, because you've got to divide it by 3, because if you've got 153 here, then you'll have 51 of these made, because every 3 of these 153 makes 1 amino acid. So 51 of these amino acids would join together to make the insulin, and there would be a gene that would control that. A gene would be the whole full length of these that codes for that, the making of that insulin. The next thing we want to talk about is how DNA controls the cell function by creating proteins. Now, one of the most important proteins it makes is enzymes, and enzymes control the reactions of the cell. So whatever we're talking about, enzymes control it. So they're one of the most important things to make. And because enzymes are made by the DNA controls how that cell acts. Remember that other proteins like neurotransmitter receptors, antibodies and hormones are going to need to be made, and enzymes are going to be involved in that. Enzymes are involved in the making of these other proteins. So enzymes are very important proteins to make, and depending on what enzymes are made by the DNA determines the function and structure of the cell, and in fact the whole organism. So the DNA is very, very important. Okay, objective G is to explain how protein is actually made. Now, DNA remains in the nucleus, and yes, it will make proteins there, but some proteins are needed elsewhere in the cell. So the DNA will have the code for it, but it's needed elsewhere. Luckily, the cell will have ribosomes. Now, ribosomes are involved in protein synthesis. They're tiny organelles dotted around the cell. So these guys can do it, but we have a problem because the DNA is so huge. How does it get its message of, yo, can you make this protein to the ribosomes? Well, it uses messenger RNA, or mRNA. Now, it's called mRNA because it's used as a messenger, but it's not deoxyribonucleic acid like this is, deoxyribonucleic acid. This is just ribonucleic acid, so it's a little different. And it's used to transfer the instructions from the DNA from the nucleus to the ribosomes. It's only made of one strand, and it's usually a lot shorter than DNA. Hence, it's really easy just to transport through the cell. These are the bases it has. It's very similar. It's got C, G, A, but instead of T, it's got U, which is uracil. And you may need to know that there's just that one base. The rest of them you don't need to know. You just need to know the letters. So what happens is, basically, the DNA in a cell is going to start unwinding, and you can see it unwind here. Okay, starts unzipping, and then 3 nucleotides. Nucleotides are these things here that we talked about, and these are nucleotides. They're going to start grouping together and forming on one of the strands. Now they match up just as normal. T will go with A, C will go with G. However, when there's an A, U goes with that, the new guy, U. So it makes a small chain of messenger RNA that's kind of complementary to the DNA that was already there. Okay? And then what's going to happen is this short chain of mRNA is going to slip through one of the pores in the nucleus. So this is a nucleus, not a cell, it's a nucleus. The cell structure is all the way out here, okay? And this is a pore in the nucleus. The nucleolus is the central part of the nucleus. So the mRNA will leave from here through a pore and then make its way to a ribosome. When it gets to a ribosome, we can just look at this one. This could be the little short chain of mRNA. It will feed through the ribosome, and as the ribosome reads the CAT, it will make valine. Obviously, it won't have that T there. It'll have a U instead of the T, but the ribosome will decode it anyway and make all these amino acids and eventually the protein that is required. And depending on where in the DNA structure, because it's huge, the mRNA starts to code from, you'll make different proteins. And the ribosomes are the organelle that do that. Okay, next thing we're going to talk about is gene expression. Now, it's very simple. Basically, every cell in your body, apart from your gametes, is going to have 46 chromosomes, and it's going to have a lot of genes. All the same genes are in every single cell, but not all of them have to be active and work in each cell. Not all of them need to make everything in every single cell. Your cells act differently. For example, your stomach cells need to make enzymes. Your bone marrow needs to make hemoglobin for the red blood cells. So all cells are going to have the genes, but not all of them have to work in every cell. And you call that being expressed. If you're expressed, you're a gene that's turned on. So you've been turned on, so you're active, and you're going to actively code to make amino acids and certain proteins. But if you are not expressed, like for example in the stomach cell, the genes that make hemoglobin will not be expressed. They'll not be turned on, so they won't make hemoglobin in the stomach cells. That would be faulty. Next one's very easy, defining haploid and diploid. We've done this before. A nucleus containing a single set, 23 unpaired chromosomes like your gametes. That's your haploid cells. Diploid is every other cell. It's the normal cells. Even the cells that create the haploid cells, like the sperm-producing cells and the ovum-producing cells, they're all 23 pairs of chromosomes, 46 cells. Somatic cells, diploid. Gametes, haploid. So we've got to look at the properties of diploid cells. So different species have different amounts of chromosomes. Humans have 46 in each cell, mice have 40. Now chromosomes in your body always arrange in pairs, one from each parent, and they're called homologous chromosomes. Say it how you like as long as you spell it right. Now it's a pair of chromosomes, one from mother, one from father. Here you go, here's a chromosome. This is the one from the mother. Here's a chromosome. It's from a father. Together, they are homologous pair of chromosomes. Now I get very confused because I look at that and go, right, I know this is a chromatid. I know that's a chromatid. I know this is a chromosome. I can see that this is a chromosome and this is a chromosome. And that's the homologous pair, one from mother, one from father. But then I look at this and go, well, hang on, that's just a single one. Will there be a centromere here to join these two together as an X? No. This is a chromosome from the female parent. This is the chromosome from the male. Okay? This is a homologous pair of chromosomes. However, the difference is here, this has duplicated. This is a duplicated chromosome. This is what they look like when they duplicate. Just like here on the karyotype, you can see that the chromosomes have duplicated. The reason we look at chromosomes like this is because they're easier to see when they've duplicated, because they're a lot bigger and they've condensed, so they stand out more. And they duplicate just before the cell divides. The cell would divide during mitosis, so this is a duplicated chromosome, and it will, one part of it would go one way and one part would go the other way when the cell divides, so that the chromosomal number is not halved. So essentially, the chromosome number in a cell goes from 46 up to 92 very quickly, very sharply, and then back down to 46 again. So essentially, the chromosome number in a cell would be 46 when they look like this. Homologous pair of chromosomes, mother, father, gave this to you. This is in your cell. This is just one pair. You have, 22 other pairs. And when the cell is about to divide, they will duplicate to look like this. So they've each got an extra bit on them now. And so the chromosome number has gone from 46 to really, really quickly 92. And then what will happen is the cell will divide by mitosis, and these will be split up again. So that will go one way with one of these, and that will go another way with one of these, and create this, which is your homologous pair of chromosomes. Now, I don't know which one you're going to get to look at in the exam, but this is a chromatid. This is a chromatid. It forms a chromosome. These are homologous chromosomes because they match each other. This could be chromosome 1, and this would be the pair that goes with chromosome 1. The thing is, the thing about homologous pairs of chromosomes is you get genes in the exact same locations. They're very similar. They have the same genes in the same order, but their alleles are going to be different. Like here, the allele is a red option and a green option, orange option, red option. So homologous pairs are very, very similar. Genes, locations, but the alleles are different. This is just a little picture here showing some DNA for some chromosomes, for some cells that are about to divide. Notice that the chromosomes are all condensed so you can see them. And this is the human with 46. Kangaroo's got 12. Blah, blah, blah.